Hello everyone, welcome to my tutorial. We'll discuss industrial microbiology and today is the introductory part. So let's start the session. Now first we have to understand what does the industrial microbiology means. Industrial microbiology is a special section of microbial biotechnology where different beneficial microbes are utilized for the welfare of the human being. So definitely these microbes are producing some compounds, some proteins, maybe vitamins or several other chemical compounds which have the beneficial utility to human beings, and we are utilizing these microbes for producing such compounds in industrial scale. So simply this is the study of the different microorganisms. Definitely those microorganisms are beneficial for their application in different industries. More precisely, this is a branch of microbial biotechnology that utilizes several beneficial microorganisms that I already told to produce different useful or high value chemicals or materials in a large scale and mass quantities for the human welfare. So what are the different industries where the application of the microbes are seen? There are mainly 6 to 7 industries like pharmaceutical industries, food industries, cosmetic industries, enzyme industries, construction industries, and definitely the production of the green energy is a completely new and have a high hype. So in pharmaceutical industry, antibiotic industries, drug industries, essential oil production, factories, vitamin production, bioactive compound productions are mainly as main aspects of these pharmaceutical industries. Within the food industry, bakery, brewery and fermentation industries are main. So let's know why these microbes are so popular in industrial applications. Among other biological organisms, microbes are extremely small in size and that make them easy to handle. This is the primary cause for this popularization. Second is the high reproduction rate. It's a very important. High reproduction rate means generation time is very short. So when the generation time is very short, then the bacterial population will grow faster, and within a very short period of time we can get a very large amount of biomass. So biomass is very much important for the industrial application because we have to produce a large quantities of the substances within a shorter period of time. Third point is can withstand a wide range of environmental variation. So they have a very high adaptability or adaptability rate. Fourth is the metabolic flexibility. Yes, microbes have different flexible metabolic reaction, so we can very easily manipulate or modulate those metabolic activities for our desired products. Easy to incorporate mutation to increase production of the essential target compound. Yes, this is another point, but this is not very easy as I read it easily. The incorporation of the mutation is easy, but incorporation of a targeted mutation is not easy, and without the targeted mutation we cannot get the desired product in an increased rate. So there are several methods of the targeted mutation that is out of the scope of today's discussion. We may discuss the targeted mutation in some other tutorials. The last point is the genetic manipulation and the production of the genetically modified microorganism of GMMO. But these also have several restrictions and regulations for production of the GMO microbes and their utilization. But these are the main points why the microbes are so popular in the industries. Next, what are the major microbes for the industrial processes? There are several innumerable, but there are some flagship microbes which are very popular, and we can give the examples of those. For example, many microbes sustain extreme hostile situation. These microbes are called extremophiles because they are inhabiting in an extreme environmental condition. And these extremophiles can be standardized for the industrial processes. Most of the extremophiles are grouped into archaea, many of them. And what are the very common examples of the utilization of the extremophiles for the industrial application? Tagged DNA polymerase. You all know tagged DNA polymerases are utilized for the PCR applications, and these are produced by the thermophilic archaea Thermus aquaticus. So Thermus aquaticus is a thermophilic bacteria that produces the heat stable tagged polymerase, tagged DNA polymerase. And another is the PFU polymerase, the same DNA polymerase which is also utilized for the PCR application. The main difference is PFU polymerase have the greater or better proofreading activity, and this is produced on another hyperthermophilic archaea, Pyrococcus furiosus. So you all know this tagged DNA polymerase and PFU polymerase have a wide application in the molecular biology. Archaea are also popular in production of the antibiotics, and most of them are called the archaeocene, and many bacteria and fungi are also involved in antibiotic production. For example, streptomycin produced by Streptomyces griseus, penicillin produced by the Penicillium chrysogenum. And many microbes are also produce different amino acids which are regularly used in our daily life, which are major sources of the external amino acid supplementation. For example, glutamic acid is produced by the Corynebacterium glutamicum, Brevibacterium flavum, Atrobacter paraffinians. Brevibacterium flavum is also known to produce L-lysine, and Brevibacterium lactofermentum strain number 487 can also produce L-valine. So you can see that the Brevibacterium, different species of the Brevibacterium can produce different type of amino acids. So we can easily modulate this metabolic activity for our target compound. So this is the beauty of the microbes in industrial application. Steroid conversion. This is a very important application in industrial microbial technology, where microbes are only capable to produce the certain enzymes which can convert the existing steroid, and this phenomenon is called steroid conversion. So microbes are very easily do that in an industrial scale, and this is important function of the different microbes for the drug discovery. Addition of a hydroxyl group to the basic steroid skeletal structure can lead to the production of a converted steroid with altered function. So with the existing steroid skeletal, we can develop several derivatives out of this steroid conversion. And this process is also called the biohydroxylation, and it has a wide application in pharmaceutical industries. The most well-documented steroid conversion is regio and stereo selective conversion of a oriented C14, carbon number 14, by different fungal microbes like Thamnostylum piriforme, Mucorbraciosinans, Actinomycor elegans, and Zygodesmus sp. So this is a very important technology where microbes, particularly fungal microbes, are utilized for the industrial application. We can discuss this steroid conversion separately in a different tutorial. Another important thing is extracellular polysaccharide. Many of you probably know the EPS or extracellular polysaccharides. Some of the bacteria can produce the polysaccharide material which are released from the bacterial cell, and they're accumulated outside the cell. They're extracellular in nature, and these polysaccharides are produced by some microorganism and released by them outside the cell, and they produced an amorphous material constituted with these elements. And they're also called the extracellular polymeric substances, or EPS, or extracellular polysaccharide, the same thing. So this EPS has varied application in different industry. Particularly, they have some certain features, like they are polymeric, and these polymers may be homopolymeric or heteropolymeric. They have a high molecular weight, 10 to 1000 kilodalton. They're completely amorphous in nature. That means they don't have any specific morphological structure. And most importantly, they may be immunogenic or immunomodulators. What does the immunogenic means? Immunogenic means the substance which can able to instigate the immunogenic reaction or immune system of an organism is called the immunogenic. So the immunogenic nature of the EPS leads their application in different pharmaceutical industry. One thing is that EPS is present in different pathogenic bacteria. Why? Because this EPS is meant for their virulence, and these factors. So one may be called that why these toxic or these non-beneficial pathogenic bacteria are utilized in industrial applications, because this EPS, in one hand, it is responsible for the virulence of the pathogenic bacteria. On the other hand, if this EPS are separated from the bacterial body, they can be utilized for the production of the antibody or antimicrobial substances which can be utilized against that particular pathogenic bacteria. So this is why the EPS are utilized in different industrial applications. At the same time, the modification of this EPS can lead to different molecules which can be utilized as a immunomodulators. So what are the function? Like pharmacological, as I already told, as a immunomodulator, anti-cancer application, anticoagulant, antithrombotic, several applications are there. This EPS, some EPS may not be that much toxic. They can be utilized for the cosmetic application, nutraceuticals, herbicide and insecticides, food technology, bioflocculant, etc. Some EPS producing bacteria are Acetobacter pochinensis, Leuconostoc pseudomesenteroides, Xanthomonas albulinians. There are several other. So this can also be discussed in separate tutorial also, because this is separate. There are many things to tell about the EPS. And another method why the microbes are utilized in the industrial microbial application is the degradation of the hydrocarbon. It's a very prominent characteristic feature. The major environmental pollutants are constituted by the hydrocarbon contamination from the petroleum industries. Massive population outburst along with increased petroleum uses makes the situation even worse. Generally, decontamination technologies, for example incineration, chemical treatments, etc., are very much costly and have limited effectiveness in case of the large amount of contaminants. And another thing is that the application of this existing technology in different location is also difficult. For example, if you have a oil spill in a large water body, how can apply the incineration of the chemical treatments there? So it's very difficult. So biodegradation of such pollutants by using different hydrocarbon utilizing bacteria are gaining popular day by day. So these there are some bacteria. One is very famous, you all know, the Pseudomonas putida is a bacteria which can utilize the hydrocarbon as their metabolic substances, and they degrade the hydrocarbons from the petroleums to produce carbon dioxide and water and different non-toxic byproducts. There are several others, for example, Achromobacter, Acinetobacter, Alteromonas, Enterobacter, Coccuria, and some members of the Staphylococcus, Streptococcus, Streptobacillus, etc. All are there. So how they act? Actually, these bacteria, by their own metabolism, they do nothing. They just utilizes the hydrocarbon for their metabolism, normal metabolism function, and for that this hydrocarbon degradation takes place into some non-toxic byproducts and carbon dioxide and water, and these non-toxic byproducts along with the bacteria itself is utilized for the different industrial application. So lastly, we have to know what are the basic scheme for industrial production or industrial application of the microbes. First of all, we have to identify a useful microbe. So identification is very much important. And second important thing is production of the pure culture. Pure culture means a culture of the bacteria containing a single population, single strain of the bacteria is called the pure culture. It's very difficult to produce the pure culture from the environmental sample, but it's not very difficult. It's a regular course of laboratory functions within a microbiology laboratory. And nowadays another technique is utilized, that's the metagenomic approach or metagenomic sequencing, where a group of different population of the microbes present in a environmental sample or any sample where the mixed population of the microbes are there, that they can be screened all together without go for a pure culture, and then by this we can construct a metabolic library and identify the specific enzyme or compounds which are our target compounds or target enzyme, and that may be called the enzyme candidate or compound, any compound candidate compound. Then we standardize for the laboratory testing or laboratory scale testing for the production of that particular compound, and then we scale up by pilot scale testing to biocatalytic application to industry by constructing different bioreactor or microbial cell factories specific for that particular bacteria. So this is a general basic scheme for the industrial production of a certain compound utilizing a useful bacteria. So thank you for listening. We'll come with another video with different topic soon. Thank you.